Mannan-binding lectin. Mannose-binding lectin MBL, also called Mannan-binding lectin or Mannan-binding protein MBP, is a lectin that is instrumental in innate immunity as an opsonin and via the lectin pathway. Structure: MBL has an oligomeric structure 400-700 kDa, built of subunits that contain three presumably identical peptide chains of about 30 kDa each. Although MBL can form several oligomeric forms, there are indications that dimers and trimers are biologically inactive as an opsonin, and at least a tetramer form is needed for activation of complement. Genes and polymorphisms. Human MBL2 gene is located on chromosome 1011.2q21. Mice have two homologous genes, but in human the first of them was lost. A low-level expression of an MB-lone pseudogene 1 MB-lone PON was detected in liver. The pseudogene encodes a truncated 51 amino acid protein that is homologous to the MBLA isoform in rodents and some primates. Structural mutations in exon 1 of the human MBL2 gene at codon 52 arg to size, allele D, codon 54 gly to ASP, allele B, and codon 57 gly to GLU, allele C, also independently reduce the level of functional serum MBL by disrupting the collagenous structure of the protein. Furthermore, several nucleotide substitutions in the promoter region of the MBL2 gene at position 550H/L polymorphism, 221X/Y polymorphism, and 427, 349, 336. del 324 to 329 17 plus 4p/. Both the frequency of structural mutations and the promoter polymorphisms that are in strong linkage disequilibrium vary among ethnic groups, resulting in 7 major haplotypes: hypa, LICA, LIPA, Alexbia, LIP, LIC, and HIPT. Differences in the distribution of these haplotypes are the major cause of interracial variations in MBL serum levels. Both HIPA and LICA are high-producing haplotypes, LIPA intermediate-producing haplotype, and Alexbia low-producing haplotype, whereas LIP, LIC, and HIPT are defective haplotypes, which cause a severe MBL deficiency. Both MBL2 and MB-lone PON genes has been repeatedly hit throughout evolution of primates. The latter silenced eventually by mutations in the glycine residues of the collagen-like region. It has been selectively turned off during evolution through the same molecular mechanisms causing the MBL2 variant alleles in man, suggesting an evolutionary selection for low-producing MBL genes. Posttranslational modifications. In rat hepatocytes, MBL is synthesized in the rough endoplasmic reticulum, while in Golgi, it undergoes 2 distinct posttranslational modifications and is assembled into high molecular weight multimeric complexes. The modifications produce MBL in multiple forms of slightly various molecular masses and pI from 5.7 to 6.2. Proteolytic cleavage also resulted in removal of the 20-odd N-terminal signal peptide, and hydroxylation and glycosylation were also detected. Some cysteine residues can be converted to dehydrolinone. Function: MBL belongs to the class of collectins in the C-type lectin superfamily, whose function appears to be pattern recognition in the first line of defense in the pre-immune host. MBL recognizes carbohydrate patterns found on the surface of a large number of pathogenic microorganisms, including bacteria, viruses, protozoa, and fungi. Binding of MBL to a microorganism results in activation of the lectin pathway of the complement system. Another important function of MBL is that this molecule binds senescent and apoptotic cells and enhances engulfment of whole intact apoptotic cells, as well as cell debris by phagocytes. Activation: the complement system can be activated through 3 pathways: the classical pathway, the alternative pathway, and the lectin pathway. One way the most recently discovered lectin pathway is activated is through Mannose-binding lectin protein. MBL binds to carbohydrates, to be specific, D-mannose and L-fucose residues found on the surfaces of many pathogens. For example, MBL has been shown to bind to yeasts such as Candida albicans, viruses such as HIV and influenza A, many bacteria including Salmonella and Streptococci, parasites like Leishmania, SARS-CoV-2, complexes. MBL in the blood is complexed with bound to a serine protease called MASP-MBL-associated serine protease. There are three MASPs, MASP-1, MASP-2, and MASP-3, which have protease domains. There are also SMAP, also called M19 and M44, which do not have protease domains and are thought to be regulatory molecules of MASPs. MASPs also form complexes with ficolins, which are similar to MBL functionally and structurally, with the exception that ficolins recognize their targets through fibrinogen-like domains, unlike MBL. In order to activate the complement system when MBL binds to its target, for example, mannose on the surface of a bacterium, the MASP protein functions to cleave the blood protein C4 into C4a and C4. The C4 fragments can then bind to the surface of the bacterium and initiate the formation of a C3 convertase. The subsequent complement cascade catalyzed by C3 convertase results in creating a membrane attack complex, which causes lysis of the pathogen as well as altered self in the context of apoptotic and necrotic cells. MBL/MASP-1 complex also has thrombin-like activity. Thrombin clots fibrin to initiate blood clots. Mice that genetically lack MBL or MASP-1/3, but not MASP-2/SMAP, have prolonged bleeding time in experimental injury models, although mice are seen to be normal if there is no insult to the body. Clinical significance. It is produced in the liver as a response to infection and is part of many other factors termed acute phase proteins. Expression and function in other organs were also suggested. The three structural polymorphisms of exin-1 have been reported to cause susceptibility to various common infections, including meningococcal disease. However, evidence has been presented that suggests no harmful effect of these variants with regard to meningococcal disease.